Okay, Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera everyone. Good morning. Salam Jumaat, salam Ramadan, 20 Ramadan kita hari ni. Today we're going to continue with aerobic and anaerobic metabolism. But as usual, this is what I'm going to remind you. If throughout the session you feel like lethargic, sangat letih, fatigue, mental fatigue, mata tak boleh nak tengok screen, dah screen time you dah habis gas dah, just stop. Don't push yourself in terms of like nak dengar juga. Tak perlu buat benda tu. Kalau tiba-tiba malam tadi you qiyamulal daripada pukul 12, pukul 1 pagi sampai pukul berapa, you rasa ngantuk ke apa kan, don't sleep in front of the laptop. Kesian laptop bukan bantal. So you just like isi borang attendance, just mention that you go only half through of the session, and then you just pergilah tidur, pergi ke apa buat apa nak dera-dera diri, nanti you catch up balik. But you're gonna miss lah. I think kalau kita ada activity, you're gonna miss that. Tapi you can catch up balik lah. You tak boleh involve je lah. Sama juga, stay like 1.5 ke macam tu, 1 hour and a half ke, as long as you can. Kalau misalnya kata you dah tak boleh, dah tak ada bateri, low bat, go. Jangan tunggu buang masa je. Nanti bila you dah fresh, you datang balik. You ganti balik jam yang you dah tinggalkan, and then you ganti balik. Itulah the beauty of online. Boleh catch up balik. Tapi kalau face to face tu, macam mana nak catch up balik kan? Susah pula. Itulah gunanya online tu. I'm doing this because saya tak nak nanti hilang keredaan, hilang keberkatan tu. Waktu kita belajar, kita tidur. Hilang keberkatan belajar tu nanti. So saya tak mahu benda tu berlaku. So I'm giving you the opportunity supaya kita punya sama-sama kerja kita berkat. Gitu. Alright. So let's share kita punya slide. Sekejap ya. Slide. Screen 1, screen 2. Screen 2. Okay. Semua boleh nampak Sekejap, saya bawa dulu chat box tu ke my first screen supaya siapa yang chat tu bolehlah tengok. Saya tengok. Okay. Sekejap ya, give me 1 second nak edit ni. Alright. Okay, tahniah kepada Hadi ya. He's doing a real good job. Saya pun tak nampak siapa masuk apa tadi. Okay, let's do the slideshow. For this one, kita akan tengok quickly part 2. Pendek je part 2 ni. We'll do some activity later on. Just to remind you again, this aerobic metabolism is super important because in biochemistry, it's all about understanding what happens in the body. What happens in the body, how we can do things. Like surprisingly, we don't eat from dawn to dusk, dusk to dawn, dawn to dusk, from dawn to dusk, and then you still can, you manage to do things. We manage to walk, we manage to think, we manage to move around, we still manage to do it without eating and without drinking. So what makes the body is so adaptable to this kind of situation? Saya ingat dulu masa tu pergi ada kerja sikit lah dekat Brazil. So we went there sebab saya nak sangat tengok Grand Mosque dekat Brazil tu. Is about 10k away from where I'm staying. So waktu tu saya tak ada kenderaan, so saya jalan. So like pagi ambil breakfast and then you jalan pergi, it's quite far away, and then balik ke my penginapan, and then kerja and then dia jalan, bila jalan tu you tahulah kat sana tu it's quite hard to dapatkan halal food compared to kalau you pergi Switzerland senang. Kalau misalnya dekat Belgium ni adalah, tapi not that as abundance as kalau dekat Switzerland ni. Memang ada lah. So saya pun kurang lah makan, tapi ada output because I walk pelan-pelan je, jalan nak tengok sana sini kan. Beautiful tempat kat Brazil tu. So pergi tu tengok coklat, berhenti juga makan coklat kan. But then I can still walk for that, pergi balik tu dalam 20k. Pergi balik dalam 20k and still can walk and still have that energy. Bila balik pun still have that energy. So where did I get that energy? So it might be from the breakfast I took, and it might be when I took my breakfast, I take protein and carbohydrate, lipid of course ada, and carbohydrate, and that carbohydrate kita simpan dalam kita punya liver. We keep it in our liver as well as in the muscles kan. So that gonna be a constant supply to me, to my body, so that I can walk pelan-pelan. Lagi satu, saya bukan go for high intensity. I still have my glycogen intact in my muscle cells and as well as in my liver. Saya still ada. Because saya jalan pelan-pelan low intensity, it will burn my fat. It will burn my fat. So badan ni seboleh-boleh dia nak reserve this glycogen. Because kalau if we do some high intensity activity, dia akan boleh ambil ni cepat. So dia akan start burning my fat. I am so gembira. So bila once I stayed there for like 2 weeks, berat badan turun dengan saya sangat gembira waktu tu. Bila balik Malaysia, naik balik. Itulah cerita dia. Itu cerita nak highlight bahawa it depends on what activity that we do and how the body tap from which resource. Kalau masa kata nutrients tu, it can come from kita punya storage of glycogen. It can also come from kita punya storage of lipid dalam kita punya adipose tissue or myocyte kan. Lipid simpan pada in our adipose tissue and disimpan pada mana? Myocyte. Dua tempat tu. Kalau glycogen, where do we keep it? Where we store it? Either in the muscle cells or in the liver. Kalau simpan kat muscle cell ialah untuk kegunaan muscle. But for liver is where when liver tengok ataupun ada trigger from glucagon, hey, low blood sugar level lah in the blood. So liver akan supply kepada the constant untuk apa lagi? Homeostasis dia lah pada kita punya blood cell, dalam blood plasma tu. Okay, and then kita ada minum air, and then we do your breathing, the oxygen. So these are all that help in as a resource or source for energy generation. Energy generation from that nutrient too. And remember that the nutrient, one that we ate, of course kalau kita ambil plant from rice ke, from wheat ke, gandum rice ke, all those corn ke, these are all plant that contain high in starch, where the starch is built up through the process of photosynthesis. So photosynthesis ni of course they require matahari to come up with that photon pack into the chloroplast, so that there'll be energy for the plant to build up that glucose molecule. So tenaga tu datang daripada photon pack of from the sunlight. Meaning that starch apa semua tu dia store tenaga daripada as chemical energy and get that tenaga from the sunlight. So therefore we can make those things as a source for energy production so that we can do work. But before it can become that energy itu, sebelum from the resource nak pergi menghasilkan energy itu, we have to go through few things. Nak bunyi ringkas nampak few things. But we are going through some complex and yet amazing orchestrated metabolic pathway and network. Dia berlaku banyak benda serentak tau. Bukan glukos saja, protein saja tak. Dia bergabung semua. Everything related. So at first when I got this paper, saya pun pening juga macam mana nak integrate everything in one idea to get everybody understand that hey, carbohydrate does not work in one silo. It does not work. It's not segregated from protein or segregated from lipid. Dia tak, ataupun vitamin tak tahu. Dia coordinated, well coordinated. Once you look at how well the coordination in the body, in our body that we cannot see in our body, you start questioning, macam mana boleh jadi macam ni? Who created it? That is for me the most important question. Who created whatever happened in the body ni? And what makes the things are regulated? Okay, kita akan lihatlah how our body, your body and mine produce energy through the process of Krebs cycle ni. Macam mana Krebs cycle tu can help in the energy production? Kita nak kena describe sikit. It's quite straightforward. Nanti my friend will explain that one to you, the Krebs cycle too. I'm asking my friend to help you to explain all those molecular level for the Krebs cycle. You'll love it. The way he explained, you're going to use more like visual notes to help us to understand. And then oxidative phosphorylation. When we talk about oxidative phosphorylation, kita ada dua komponen kat situ Ada ETC, mana pen? Where's my pen? ETC and chemiosmosis or proton motive force. Nanti kita tengok yang tu. Also, my friend will do that for me, on behalf of me. And then kita can go for description of electron shuttle system. I will do that. And quickly on the regulation and do the comparison of aerobic and anaerobic metabolism. Generally, you guys already got that idea, the comparison, but we're going to look into more into aerobic and aerobic nanti. Okay. Here again, to get that principle, sama juga macam saya nyatakan tadi when we have food where we digest, and then kita ada metabolism, where this is where the body will pick nak breakdown ke, nak build new molecules ke, ataupun ada pathway yang dia ada both, which is known as metabolic metabolism kan. So all of this for catabolism is to supply for the process of oxidative phosphorylation, to supply that NADH2, FADH2, nak supply that to the oxidative phosphorylation. For what? To produce ATP. That is the major mission is to produce ATP. Nak produce ATP ni, dia ada proses dia. We have the oxidation, where it comes from the word oxidation. Oxidation will happen. Kat mana oxidation to happens? It happen in the ETC, where we have NADH or FADH2 will lost their electron. And there'll be a passing of electron through that ETC, electron transport chain. So under oxidation reduction, other those process. So at the end of it, when those oxidation reduction process, the electron will be passed on from one complex to another complex. Yang tu kita dah tahu kan. At the same time, there'll be a pumping out of this hydrogen ion out to the intermembrane space. So nanti akan ada lah bila push it out, we can observe the difference in gradient, in the concentration gradient. Ada gradient of concentration pada hydrogen ion tu. So apa yang akan berlaku kat situ, in terms of making it balance balik, they have to go through a protein channel. This protein channel ni nanti, they kind of like turbine. So they can spin macam tu, ada some sort of way of spinning it. At the same time akan berlaku phosphorylation. Lepas tu kita ada perkataan oxidative and phosphorylation. Phosphorylation ni happens pada ATP synthase complex tu lah. So kita add phosphate kepada ADP. So that is the phosphorylation to produce ATP. However, this one, the NADH ni comes from various sources ataupun various resource. It can comes from glycolysis. It can also comes from the breakdown of protein, the breakdown of fatty acid, and also in the Krebs cycle ataupun TCA cycle. You boleh sebut mana-mana pun boleh. Selalu saya cenderung sebut TCA cycle ni sama je lah. Tricarboxyl citric acid cycle ni, ataupun you can use Krebs cycle pun boleh. So it can come from various resource atau various source kat sini. However, for glycolysis nanti, before it can go into that mitochondria, into that matrix of mitochondria for further production of ATP, there'll be some changes. There'll be a shuttle that to bring in the energy from NADH produced from glycolysis. We're going to look into that later. And finally, kita will go quickly to understand, ada benda yang regulate, to identify to identify what regulates all those metabolism. Ada yang enzyme, kita panggil ni, biasanya kita panggil ni control point lah. Control point tu dia macam checkpoint yang kita boleh manage. If you recall kita punya enzyme punya discussion last time, ada yang dipanggil allosteric kan, allosteric site. We talk about competitive inhibition, we talk about uncompetitive, non-competitive. We also talk about the allosteric site. So these are how the enzyme can be activated or can be stimulated, can be activated. As well as hormone. Hormone juga play a very important role to regulate certain pathways. Gitu. Itu important principle yang we need to understand on this aerobic and anaerobic metabolism. Later on, kemudian ni habis yang ni, so minggu depan, kita dah start zoom in into what happens in carbohydrate, what happens in protein, what happens in lipid. Dan whenever we discuss about that, kita kena link balik how it supply dia punya NADH and FADH to that for the process of oxidative phosphorylation. Gitu. Okay, quickly to get the idea of how those things are related, it looks as a very complicated, but this is only visual saja. Ini visual infographic sajalah. But then kita tak tengok semua for biochemistry. We look at the carbohydrate metabolism. We look part of lipid metabolism. Tak semua tau. We look at sedikit bahagian for amino acid metabolism. And what else? Kita tengok, yeah. 3 tu je lah for principle in biochemistry. But there are lots of things. Other than that, kita ada cofactor and vitamins metabolism, all those things. Ada banyak lagi yang for me it's amazed me. Kalau misalnya kata you are on the same page, nanti you boleh venture, do your master level and study more about any part of that dan yang kita minat, yang you minat nak buat. Macam saya dulu, my master degree is just to look at the photosystem tu, just to look at dalam plant kan, dia ada photosystem tu ada satu bahagian dipanggil the complex. Saya tengok, saya block dia punya electron transport. Saya block dia punya electron transport and turn that into a biosensor. Itu saya buat dululah. But then you might want something else other than that. Nak buat benda lain, tutup pulang lah. Okay, ini just to recall balik kita punya discussion last week. Mainly is to get the oxidative phosphorylation because we want to produce more ATP. Itu the purpose lah. And end product tu of course kita ada eliminate of ammonia situ, kita ada production of air, kita ada carbon dioxide, and as well as kita punya energy. Yang tu yang kita nak to get those energy. So that is the idea of biochemistry. Nak tahu macam mana boleh dapat energy tu towards the end. From carbohydrate, from protein, from lipid. Macam mana nak dapat tenaga tu? So that we understand the body and then you know how to deal with your life lah. Kita punya health kita tahu macam mana nak buat. Talking about health, jangan lupa minggu 5, hujung minggu ni just collect your data. Ada juga yang belum collect data tu, seram. Okay. We talk about aerobic metabolism, so we should also talk about the anaerobic metabolism. Last week, you guys did very good, super quick in identifying a good infographic to do the comparison between aerobic and anaerobic metabolism. As we know, kita ada beberapa jenis lah anaerobic respiration. Contoh, we have lactic acid in animals kan. Kita ada lactic production of lactate toward the ends of the process. We also have the one in alcoholic fermentation dalam plant contohnya, yeast contohnya. So maybe ada some fish juga macam contoh goldfish. Siapa yang bela goldfish tu, berarti dia pun boleh melakukan alcoholic fermentation. At the end, dia akan menghasilkan ethanol. So, other than that, kita ada propionic acid fermentation, so on and so forth. These are various types of fermentation. But for kita punya pembelajaran, there's part that we need to look into. Kita selalunya kita akan rujuk, of course kita akan rujuk, sekejap saya stop sharing dulu. Kita akan rujuk dokumen yang diberikan oleh kurikulum. Kurikulum sekolah lah kan. Itu semua mesti dah mahir. I hope that semua orang pun dah berjinak-jinak, is that a good word? Ataupun get yourself familiarized dengan buku teks. Misalnya kata if you are majoring in chemistry ke, physics ke, math ke, geografi ke, pendidikan awal kanak-kanak ke apa ke, get yourself familiarized. Waktu dalam tahun-tahun ni jangan ujung tunggu ambil KPD baru nak buka buku teks. Betul tak? You tak tahu apa benda yang berlaku dalam buku teks tu. Because nanti you become the master of buku teks. And for your information, government is looking forward. I think they are planning to build college tingkatan 6. The first college tingkatan 6 will be in Kelantan. So you bila once you dah graduate, you memang dah ada lesen mengajar kat situ. So make sure that you know what is college tingkatan 6 punya syllabus. It's sama lebih kurang macam matrix jugalah. Sama juga if you want to go for College Mara, College Mara ada banyak foundation. So dia punya content pun sama, lebih kurang tu je. Lebih kurang tu je. The depth is sama saja, sama saja at that level. You can also be teaching diploma. You pun boleh ajar diploma sebab you ada degree nanti. Okay, if you look at dalam buku teks Form 4, this is Form 4. This is what the curriculum KPM yang come up with. Who knows, nanti you pun tentang nanti kerja dekat ni kan, KPM bahagian pembangunan kurikulum tu. Okay, so let's look at apa benda yang dia fokus. Saya scroll sekejap. Scroll pula. Apa lagi? Zoom in sekejap. The production of energy through cellular respiration. You ada buku teks, you buka. Tengok, dia pun nak tengok juga Form 4, they want the student to be able to list types of cellular respiration. Aerobic ke, anaerobic ke, and also fermentation. Dia letak 3 kat situ. Dia tak letak anaerobic saja. Dia ada satu lagi, dia letak fermentation. So ada 3 dia punya cellular respiration. Macam for biochemistry kita tengok 2 je. Because fermentation nanti you akan tengok pada subjek lain. Tapi of course it will be pengenalan lah kat sini. And then apa? Necessity of energy, very simple idea. If I can ask you now, I think all of you be able to answer why we need justify the necessity of energy in metabolic processes. Kenapa perlu tenaga dalam proses metabolik? Identify the main substrate used in energy production. Semalam saya tanya group, apakah kita punya the main substrate for cellular respiration? Majority boleh jawab. I believe kalau saya tanya you apa, you boleh jawab. Apa benda yang main, yang paling utama sekali cellular respiration, substrate apa? Kalau you orang rajin nak type lah. Jawapan dia I think semua pun dah tahu. Kalau siapa tak tahu, you tembak aja betul. Kalau you tengok you tembak salah, it's okay. Nanti you betulkan. Alright. So 7.2 aerobic respiration, saya tengok. Dia nak tengok conceptualize. Menkonsepsi. Ada perkataan Melayu yang saya pun pening juga. Conceptualize energy production from glucose during aerobic respiration in cells. Once you tengok tajuk, you tak faham tajuk ni apa benda mengkonsepsi, you just go to the text. Pergi dekat text. And at this point, they need to write a word equation. Macam kita tak tengok word equation. We have to look in detail lah. Structure berapa, carbon berapa. You kena tahu lah benda tu kan. Conduct experiment. So mesti ada kemahiran. So guru mesti ada kemahiran untuk melaksanakan eksperimen. Masalahnya sekarang kita pun tak boleh nak train for nak buat lab. Tapi insyaAllah katanya you guys will be coming in secara besar-besaran tahun ni juga. So kita tengoklah, kita jumpalah di makmal nanti. So fermentation, kalau kita tengok these are fermentation, ada cerita-cerita dia kat situ. We can look at the notes. Tengok dia punya ni, aerobic respiration. They just go for glycolysis, glucose pyruvate. In between, dia tak diperkenalkan lagi. Walaupun dia tak diperkenalkan, kita kena tahu. We have to know what happens between glucose and pyruvate. Kita kena tahu apa yang akan berlaku in between. Before that, Hadi,
Ya?
Uh, is it mengganggu you ke nak fokus sebab you kena admit orang masuk?
Tak
I
ada.
can do that. I can do that.
Tak apa, tak apa.
Kesan pula you nanti tengah bau, tengah nganga tu ada masukkan tu pula kan. Patut minta tiket je lah. Siapa masuk lambat tu kita minta tiket sikit. Alright. Oxidation of pyruvate kat sini dalam chart ni dia terus tulis carbon dioxide, water and energy. Not talking about what's going on. What does it mean by oxidation of pyruvate? How can we produce air, water, carbon dioxide, water and energy? Macam mana Tapi for kita punya part, that is where we understand from pyruvate to acetyl-CoA, and then ada the Krebs cycle, then production of NADH, production of FADH2, and these will go into the electron transport system. Barulah akhirnya menghasilkan carbon dioxide, water and also energy. Macam gambar raja tadi tu. So, dia tak diperkenalkan lagi. But kita dah kena kenal So, word equation, so forth, the lab work that they have to do, and talk about fermentation. They talk about this alcoholic fermentation, yeast and plant. So these are the things that kita kena dah ready. Apa beza alcoholic fermentation dan lactic fermentation tu? Lagi dia tengok apa. Lagi for human punya lactic acid fermentation, kejap saya buat satu page. Satu page kat sini. Ni dia punya penceritaan dia. Dia punya narration dia lah. Dia buat daripada tadi tu sambung. Lactic acid fermentation happens in animals and human kan. Juga dia tunjuk cell. Lactobacillus, bacteria lactobacillus. So ini apa berlaku. Macam mana boleh menghasilkan that soury taste. Bukan soury, memang sour kan. Sour taste in yogurt tu. What's going on in there. And also in human muscle. And they also talk about oxygen debt. Oxygen debt 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 jangan sebut debt. Debt is debt. What does it mean by oxygen debt kat situ? So these are all the things that kita dah kena ada idea tu lah. So contoh I think most probably it will be explained in animal physiology. Tapi ni pun you boleh dah boleh tahu dah. You boleh tahu what does it mean by oxygen debt. It's when we do exercise, bila kita buat exercise, you tengok kat sini figure 7.1 ni. Bila kita makan pula. Bila kita do the exercise ni, you breathe in, breathe out, breathe in, breathe out. You buat rigorous, buat, and then you just lack of oxygen. That is known as oxygen debt. Ada latih, bila you berhenti, and then kita ambil nafas balik. So, and then kita bayar balik lah dia panggil kan. So, that is to show that during exercise akan berlaku oxygen debt. During recovery, kita akan kita bayar balik hutang. Cerita macam tu. But also, bukan senang dia cerita macam tu. What's going on during that exercise dia masukkan tu? Oxygen intake during exercise tu, apa yang berlaku sebenarnya? So, those are the things that we need to discover. Kita kena tahu apa yang berlaku untuk itu. Okay. Now, do you still have Padlet punya link? I think you ada lagi kot Padlet punya link tu. Ada kan? Kalau siapa yang masih ada tu, ni. I'm going to copy that. I'm going to put in kita punya chat box. Siapa yang tak ada, you just go. Kita punya task, I'm not so sure whether those are using phone could get through this. Sekarang you cari, go to Google Image, you find out the best infographic. The best infographic. Sekejap, ada ke task kat situ saya masukkan? Ada. Find the best infographic for anaerobic metabolism pathway. The one that I showed you here, I'm showing you on the screen, ialah very simple, simplified. Tapi, do we have an infographic that shows the actual things happening in anaerobic metabolism? Cuba you cari best infographic for anaerobic metabolism pathway. Okay, listen to the instruction. So find the best infographic that we can find in Google Image on anaerobic. Anaerobic bukan aerobic, anaerobic metabolism, and where that infographic can provide you with many information. So you just post it in the Padlet, and then rakan-rakan yang lain boleh screen tengok which one you feel that this one is useful, and then you click like. Yang mana you rasa macam ini useful tapi tak informatif, you tak apalah, you tak payahlah like kat situ, walaupun you like dalam hati. Yang mana yang you rasa okay, this one is good, this one is giving me information. So to describe anaerobic metabolism using infographic, kalau you jumpa dekat itu tu, sila upload dekat sini. Jangan terpengaruh dengan dia punya wallpaper ni. Ni cuma rekaan semata-mata. Dia baru pukul 9. Belum boleh berbuka puasa lagi. Alright, silakan. While waiting for you to put in, I am going to stop the video and mute myself. Okay, ada minta repeat ni. Repeat apa tu? Repeat arahan ke? Repeat dia pergi dekat Google Image, cari anaerobic metabolism pathways. The best that you kata this is the most informative infographic, and then you upload dekat Padlet. Lepas tu kalau kawan-kawan lain dah tengok, dia kata oh this one is the best. Yang informatif, you like dekat kawan tu. Bukan like semua orang ni. Last week kita like semua orang. Hari ni bukan kita tak like orang tu, kita like nota tu. So like nota, this one is very informative to explain about anaerobic metabolism. Gitu. Kalau nak like diri sendiri pun boleh. You can put in your name if you want. Kalau tak nak pun tak apa. You nak letak nama pena kan. Nama stage. You punya nama artis ni pun boleh. If you want people to know who did what, pantai je lah nama kat situ. Letak je lah. Pantai pula. Okay, those who have already uploaded their infographic, some of it you can see similar, sama, same. It's not similar, same. And ada yang that you can just look around and see which one you feel that is informative, that you understand. That you understand, it makes you understand what is anaerobic metabolism is all about. Tengok kalau jumpa, you like kat situ. Mana tadi? I saw one that can give a general idea untuk siapa yang tak berapa nak faham sangat apa benda berlaku tu, mana dah hilang lah pula. I thought that is a good one. Mana dah? Well, I'm not so sure mana dah tadi because I saw yang okay. Tapi dah tak ada dah. Ada siapa tarik tutup balik ke? Delete ke tak? Not so sure which one. So if you look around and see which one that you think that can makes you understand better, apa to understand better apa benda apa dimasukkan dengan aerobic and anaerobic metabolism, aerobic metabolism and anaerobic metabolism. So just give a quick like kat situ. Yeah, I saw just one, tak sempat nak like benda tu, dia dah hilang lah pula. Kalau siapa-siapa yang tiba-tiba dia tengok dia punya hilang tu, ke saya dah terdelete ke apa ke? Okay, anyway. Oh, couldn't find it. Takkan saya delete kot. Nanti boleh tengok video. So, yeah, let's look at this one. This one ni kalau kita tengok, dia punya more like words equation Kalau you kasi this one that can makes you understand, then okay lah. Kalau siapa-siapa lah ada idea lagi, this one is glycolysis. Dia tak adalah menunjukkan, it doesn't shows the production of lactate or alcohol. So this one is not related lah yang ni Dia tak related. Yang ni, this one is a clipping actually. Kan ada dalam buku pada. So you can look at dia punya end pun dia akan tunjuk menghasilkan pyruvate. So this one ni ada tak ini related to anaerobic? I can't find any part yang tunjuk dia menghasilkan pyruvate. Sebelum dia pyruvate tu akan ada stage yang dia akan supply it to form lactate tu. It's not there. So ni this one. Lebih baik saya like siap-siap. This one tadi it's small, but to show you how the glucose becomes pyruvate, right? This is glycolysis. So pyruvate ni, in terms of lactic acid fermentation, it will produce lactic acid. In between two, NADH will pick up the hydrogen from pyruvate, will pass on the hydrogen, the NADH will lost the hydrogen to pyruvate, so pyruvate can become lactic acid. And then the NADH ni, we can go again to the process of glycolysis and help in picking up more NADH. So therefore ada net ATP kat situ. That is why when we talk about doing lactic acid fermentation, we still gonna get energy. Kita still akan dapat energy juga, but lower compared to if we go for complete oxidation ataupun kita go for glycolysis. So that's one okay juga. Yang satu lagi tu yang ni. So this one is a good one too. It can help us to understand glucose. See, glucose dia menghasilkan ATP, form it into pyruvate. So once dia dapat, it release dia punya decarboxylation. Ni more in detail. Ada decarboxylation, dia akan jadi acetaldehyde, and then NADH akan bagi lagi hydrogen kepada this acetaldehyde to form into ethanol. And then dia lagi menghasilkan coenzyme NAD+ to pick up more of hydrogen. So still boleh menghasilkan energy. That is for ethanol, but for lactic pathway is different. The lactic pathway is different. Bagi je hydrogen tu, then it form into lactate ataupun jadi lactic acid. So here is pyruvate tu sendiri akan terima the electrons from NADH. And yet still 2 ATP produce that. So overall kita tengok siapa yang disukai cahaya punya. Cahaya apa tu? Cahaya 114. So ada untuk mewakilkan lah. This is a simple infographic. Maybe this one, I think this one would be more explanatory punya structure. Ada satu lagi yang detail. Alah, mana dah? More like the structure tu. Then you can go for that one too. But anyway, congratulations everyone for be able to put in information and informative foto or infographic, but some of it yang itu bukan anaerobic. Some of it itu masih glycolysis. We cannot observe any part yang macam ni kan. Of course ini glycolysis. You cannot observe any part yang indicates ada nota ataupun ada maklumat information on anaerobic. Yang ni nampaklah ni anaerobic kan. Nampak? Tak ada. Ni tak ada. Terus tak ada. Tapi kalau yang ni, dia punya partner dia yang ni. Gambar rajah ni. Sekejap. Ni. Yang tadi tu just half of it. So ini half of it. So sini kita boleh tengok the comparison in between. This is one pun okay juga bagi like kat situ. So you can keep on viewing. And this one pun bagus. This one pun bagus. This one from Net Geo graphic. You pun boleh tengok juga ni nanti. Yang the green one is an aerobic respiration. The green arrows. The blue one, of course, yang tu ialah aerobic respiration. You can look at the different. This is a good one as well. I used to use this one for my diploma student. Tapi okay, you all pun boleh guna sama je. Pyruvate menghasilkan ATP. Nampak 2 ATP kat sini. Pyruvate nanti it will pick up the hydrogen to form into lactate, and then this is what is known as fermentation. But yet produce ATP. This lactate ni nanti dia akan go to blood lah, pergi ke darah so itulah yang orang kata the soreness come from lactic acid tu yang tu lah, but actually it's not come from the lactic acid, it's come from the potassium ion yang berlaku dalam badan tu dan yang cause soreness tu. Okay, that is for the Epic board. I'm going to download this one later on. Siapa nak masukkan secepat. Because I'm going to download it. Nanti I will put it in your Google Drive so that you nak masukkan dalam you punya e-portfolio. You punya apa benda you hasil yang dah buat, and then will be okay. Tapi masukkan cepat because at the end, lepas kelas saya terus buat. Sebab kalau siapa yang nak masuk lambat, petang ke, lepas buka puasa ke, benda tu dah upload dah. Alright, so that is the one. Now, next thing ialah, let's go to my notes tadi tu. Apa ada lepas ni? Important topics. Okay. We can do this quickly. Or, we can do this quickly. Alright. Before we go on to this one, go ahead, take a break. Sekarang dah approaching 9:30. Kita 9:35 masuk balik. Siapa yang line dia suka hilang-hilang tu tak apa. Nanti you masuk balik. 9:35 Okay, masuk balik. Kita resume at 9:35 AM. Okay? Okay, see you there. Kenapa Nur Syuhada ada anak berlari-lari? You nak pergi jogging ke? Okay, bertemu kembali. Alright, so let's review on the important topics. Kita tahu citric acid cycle. We know about citric acid cycle. We know what's going on in citric acid cycle. That it will produce an ADH and FADH2. And also other intermediates. Intermediates can be in form of, kita letak kat situ, alpha keto acid. Structure of alpha keto acid. Contohnya macam oxaloacetate, alpha ketoglutarate, those are some example lah of alpha keto acids. So and then kita tahu bahawa this NADH and FADH2 we go through the shuttles. Kita ada two types of FADH2 FADH2 straight away pergi ke situ. NADH, it depends what types of NADH. Where does the NADH originate? Dia datang daripada mana? If that is cytoplasmic NADH, it will have to go through shuttles. Kalau dia dalam bentuk mitochondria NADH, dia tak ada masalah terus ke electron transport chain. I think the graph that I made ni salah. Dia cause misconception because shuttle bukan untuk mitochondria. Shuttle ni, if I can make a one big box and contain semua tu, dia untuk cytoplasmic ni. Cytoplasmic NADH. And then we have, berapa banyak kali nak round tu. Shuttle ni kita ada dua jenis. We have, one is malate aspartate shuttle, as well as glycerol 3-phosphate. Glycerol 3-phosphate shuttle. So there are two types of shuttles. So once the all the transfer of energy molecule, the electron and hydrogen ion berlaku, so dia pun sama, will produce that NADH. Oh, salah, sorry. Confused. The shuttle ni will also go to the electron transport chain. Either it will go in form of NADH ataupun dia go dalam bentuk NADH2, FADH2. So dia akan go to electron transport chain lah. Kat situ nanti ada all those passing of electron redox reaction. Redox ada oxidation, reduction, so on so forth. At the same time ada proton pumping. And then to get that concentration balik sama in equivalent kat luar intermembrane space and in mitochondria, nanti kita akan ada the production of ATP at the end of it. Sama juga ada penghasilan water molecule and also ada penghasilan carbon dioxide at the end of the complete oxidation. So ini adalah important topics yang kita kena tahu. Saya ulang banyak kali benda ni because so that benda tu lekat dalam kepala kita semua. And here is the citric acid cycle. Citric acid cycle ni, you just can hover your mouse ke situ, nanti dia akan terus bawa you kepada my friend. Dia akan terangkan kat you pasal Krebs cycle ni in detail, the structure. But if you look at the structure, berapa carbon, take note. Carbon ada berapa? Hydrogen ada berapa? So bila ada perkataan dehydrogenase, you know there will be loss of hydrogens going on. Anything that lost electron, lost hydrogen ion, that is the process of oxidation. Anything that you can see the molecule gain hydrogen atoms or gain electron, these are the process of reduction. So dia ada selalu couple macam tu, redox. That's why some books or some reference they terus guna redox reaction. Okay, duration for this video is 32.15 minutes. You can skip ataupun you can speed up dia punya playback tu bagi cepat. Ada setengah orang dia tak boleh dengar lambat-lambat, dia nak dengar cepat. So you can just playback sampai 2 times ke, it's up to you. And then you can just go through back and forth supaya boleh nampak lebih jelas. You can repeat it, benda tu balik so you boleh faham lebih lanjut lah. For this one is to look at the regulation of glycolysis. Sama ada this pyruvate ni nanti dia akan pergi ke Krebs cycle ataupun tidak. What is the fate of that pyruvate, for instance? So we have two condition. Kalau tengok kiri kanan ni to compare. Yang ni ialah to look at rest. And this one, sibuk pula aku kat situ, ialah to look at during exercise or during kita membuat kerja, when we do work. So when we are at rest, we don't break glucose. When we are at rest, relax, ya kan? Kita kunyah makan ke apa benda tu bekerja ke? It's not that hard sampai nak kena break kan. Of course dia kena break juga. Of course kita akan guna ATP wherever in the body kan. But if we, for instance, like we do nothing, we just relax, we don't break the kita tak break pun kita punya glucose tak break. Dah ada cukup, kita tak break. So ada two ways of controlling it, to inhibit it, right? So as I mentioned awal tadi, kita ada kita punya control point. It can be enzyme, it can also be controlled by hormones, it can be controlled by both hormone and also by enzyme, by the protein, and as well as by the intermediates, as well as by the product. Contoh macam ni, the first one. Glucose in glycolysis, glucose will produce in that pathway kan. Glucose will be phosphorylated. Phosphorylated means glucose will accept phosphate group. Dia akan terima phosphate group. Therefore, kita dapatlah glucose 6-phosphate. Where the phosphate is attached to carbon 6. Sebab tu ada 6 kat sini kan. It will attach to carbon 6. Phosphate tu. That is known as active form. Inilah bentuk active form. Kalau glucose saja tu dia tak active form sebab dia boleh ke sana dan kemari. Active form. Nanti kita tengok semua pathways mesti ada bentuk active form. Nanti dia akan ready to be metabolized. So, jadi G6P. If in the body we have high amount of G6P, macam mana? Dia akan beritahu glycolysis, slow down. Slow down, right? Jadi panggil negative feedback. Dia akan help in the inhibition of hexokinase. So tak adalah lagi phosphorylation tu berlaku. So negative means the negative buttons tu, minus button tu means inhibit. Kalau positive button ni maksud dia stimulate. So itu satu. The second one is looking at the energy level. If the body ratio of ATP to AMP, adenine triphosphate to adenine monophosphate. Kalau ATP tu tinggi, berarti tinggilah. Kalau high ratio, berarti tinggi. Katakan 10/1. Of course lah, tinggilah kan. So, when ATP is high, it will inhibit two enzymes. It will help in inhibition of two enzymes. Phosphofructokinase, so that there will be no production of fructose 1,6-biphosphate, as well as pyruvate kinase. So, it will stop from producing pyruvate. Itu cara dia. So, dia tak jalan lah kita punya. That is how the body regulate. Kita tak payah pun nak cakap, okay, glycolysis, inhibit, mode on. Tak ada. So, because we have this system organized in the body. Satu. What about if we do exercise? You just go upstairs, down, up and down, you do house chores, nak raya kan, cuci berus-berus lantai, berus longkang ke apa, cuci siling ke, lap-lap ke apa benda ke, you do serious activity bukan setakat tekan robot vacuum tu tak. So during exercise, when we do work, kita jalan-jalan ke, jogging ke apa ke, so macam mana kita nak stimulate glycolysis? So when we have the body is alerted by AMP tinggi daripada ATP, meaning that the ATP is low, ATP tu rendah, kurang so dia akan stimulate PFK, phosphofructokinase, so that dalam badan yang dah ada fructose 6-phosphate tu readily will be phosphorylated again. So F6P will be phosphorylated again. Sebab tu dia ada biphosphate, 2 phosphate dan pada karbon 1 dan karbon 6. So bila dah ada pembentukan F16 biphosphate, so what happens next? Ada satu lagi, dia akan go for feed forward stimulation. Feed forward stimulation. Dia akan maklumkan awal-awal dekat pyruvate kinase, we're going to have like more PEP coming in. So you must be active. Ni PEP lah ni phosphoenol pyruvate, PEP. Dia akan alert pada enzim pyruvate kinase, hey, there'll be more PEP, so you better get ready because we want to produce more pyruvate. Gitu, cerita sembang lah kan. So that is where ada simulation, hasilkanlah pyruvate. So pyruvate, of course, you go through Krebs cycle, ETC, and finally kita akan dapatlah that end product as well as kita akan dapat ATP. So ATP akan, of course, akan dekat naik. Or kalau kita buat sprinting tadi tu kan, without oxygen tu, of course kita akan go for lactate fermentation, like the one that you did dalam you punya padlet tadi tu. Itu cerita dia, how the body react when we have high energy, high ATP, or if we have low ATP. Kalau kita tengok dua-dua ni, you duduk, you tengok, apa hubung kait ni? For at rest, ATP ni dia targeted to PFK and pyruvate kinase. But for during exercise, ATP, AMP, low energy tu, dia AMP tu lah, dia targeted satu enzim je, dia tak target dua. Dia target PFK. Because when dia stimulate PFK, they only stimulate PFK, it did not stimulate pyruvate kinase. Yang stimulate pyruvate kinase ialah the substrate, the intermediates tu, F16 biphosphate. So that is the one, the intermediates that stimulate pyruvate kinase. Cuba tengok. Yang tu dia panggil apa? Itu dipanggil study. Itulah yang dipanggil study. When we look at the difference between two situation, two event, and then we look at apa similarity dia. Ada tak similarity dia in between? Which one is functioning? Which one is not playing any role? Which one is the most important or most crucial things that can trigger or that can inhibit a pathways. Ni contoh dia lah. Contoh kita bila kita nak study. Bukan baca satu tu okey. Itu mengenali to identify the elements in the pathway. That's okay. You identify dulu, okay, elemen ni phosphoenol pyruvate. Nak sebut dulu kan. Belajar sebut nama dulu. You understand the character. Like you watch movie, you understand the character, you know the character ni nama apa, and then you look at the story. So these are the stories. Then you start comparing story ni dengan story ni. Contohlah macam you siapa yang suka tengok Spider-Man kan. 1, 2, 3 nak tukar watak-watak lain kan. Of course you can tengok which one is better. Oh, I prefer Danny Tom Holland lah. I prefer yang ni lah. Contoh kan. Because dia buat comparison. When you do that comparison, that is critical thinking. Sedar tak sedar kita sedang ada critical thinking. You do comparison, you tengok yang mana best. Apa yang sama ini? Macam mana boleh ada ni? Oh, ada multiverse. You know, that kind of idea when you look at things, that is called study. Study. Maksudnya you analyze the information that you have in hand. Okay? Cerita Spider-Man pula. Alright. So, when we talk about shuttle, maybe I think majority of you pun dah tahu cerita pasal shuttle ni. Ini mungkin siaran ulangan saja. So this shuttles happens when kita ada when we have our glycolysis that produce NADH. Saya letak C kat situ mean cytoplasmic cytoplasmic NADH. They produce outside of that mitochondria. All this ETC happens in mitochondria kan, pada mitochondria. But there will be situation where it happens, activity yang happens in cytosol, which is glycolysis. So to get that NADH into the matrix mitochondria is never going to happen. Because of NADH tak ada satu specific protein or channel to bring it into the mitochondria. Mitochondria is very selective. Macam you nak masuk C lah kan, sangat selektif. C lagi, interview lagi, nak itu lagi, and then baru boleh masuk. Sama lah juga ni. So, to get that NADH into the matrix of mitochondria, it will never happen. But, what can happen is that it can get dia punya hydrogen ni. That hydrogen from the glycolysis that carries that energy, it can be transferred. Dia boleh kirim dia punya hydrogen along with the electron lah, of course, along with the electron. They can kirim it. Nanti kalau you jawab soalan, jangan cakap kirim. It is to make us understand. It can transport it. It can transport the hydrogen along with the electron through a molecule. Through a molecule. That molecule adalah malit. That molecule adalah malit, because malit will carry that hydrogen and go through that channel, a protein channel. So for this shuttle, kita ada dua protein channel. The first protein channel is malate and alpha glutarate transporter. So the word malate and alpha ketoglutarate tu maksudnya only these molecules can go through it, either from intermembrane space into the matrix ataupun from matrix into the intermembrane space. Number one. The second one, kita ada glutamate aspartate transporter. This is also can allow glutamate and also aspartate to go in or out through that protein channel. The protein channel bukanlah color pink bulat macam tu. Dia ada structure like I showed you the saliva kan. There is special structure for it that only allow these molecules to goes through it. Okay, that's number one. The second information that we need to know is that malate, macam mana dia nak obtain that hydrogen ion? How is malate going to obtain that hydrogen ion? Hydrogen ion from NADH of glycolysis. How they are going to do that? Of course, before becoming a malate, dia ialah bentuk lain dulu. Before it becomes malate, before it becomes transporter tu, dia kena jadi bentuk lain dahulu. That bentuk lain tu ialah oxaloacetate. Ni, oxaloacetate ni. Oxaloacetate. Super important in this event. Oxaloacetate ataupun also saya kalau refer to protein metabolism, nama dia kumpulan alpha keto acid. We can find it in amino acid metabolism. We can find it in Krebs cycle or TCA cycle. Rata-rata jumpa oxaloacetate ni. It can also become a part of involved in the carrier. It can also kita akan jumpa dalam amino acid transfer kiri kanan tu, transfer in amino group tu. So let's look at the first one. So oxaloacetate will get this hydrogen ion from glycolysis tadi tu, and then if you look at the structure here, dia dah ada carry the hydrogen which comes from NADH. Very strong energy there. So because of the bond kan. So that malate goes into the matrix of mitochondria. Dia pun masuk. So sekarang remember that kita nak that hydrogen yang we nak transfer tu dalam bentuk NADH. Bukan dalam bentuk malate. So what malate can do is that they can go through an oxidation. So dia boleh go through an oxidation. Oxidation means dia akan release dia punya hydrogen dia ni. Release. So that is why kita nampak kat sini enzim malate dehydrogenase. So this is isi satu lah Kalau ingat tak ingat oxidoreductase enzyme. Kalau kita tengok ingat balik recall our enzyme discussion last few weeks. So oxidation. When oxidation happens, mesti ada reduction. So what will be the one that will accept the hydrogen? This coenzyme NAD+. So this coenzyme NAD+ will accept that hydrogen tadi daripada malate. So now that it becomes NADH. So benda ni semua akan pergilah kepada electron transport system to the next session tu lah, the next process ETS, bukan ETS Tanjung Malim KL tu so that's how it's happened. So yeah, habis cerita. So the thing is that does it ends there? Oh tak. Sebab tadi malate when it lost dia punya hydrogen ion, dia dah balik kepada, it goes back into dia punya bentuk asal, which is oxaloacetate. Oxaloacetate. Itu mission pertama dah accomplish. Mission pertama dah accomplish. The first mission dah accomplish. Second mission ialah to get this oxaloacetate keluar balik. To get that oxaloacetate keluar balik, or we have to get this oxaloacetate back to the intermembrane space. Kenapa? Because we want to pick up more of hydrogen from cytoplasmic NADH. Kita nak shuttle kan? Kita nak transport. So we need oxaloacetate outside. So what we can do here is that we can either get it through malate yang masuk tadi sini, here, but we can't because it's only meant for malate alpha ketoglutarate. But we have another protein channel atau untuk transport ni, which is known as glutamate aspartate transporter. Oh, okay. So what can oxaloacetate change into? Kalau dia terima hidrogen, dia jadi malate. So sekarang, oxaloacetate can be transformed into aspartate. It can change into aspartate, provided that oxaloacetate is supplied with nitrogen. Sebab tu, alpha keto acid. I will try to write this nicely. Amino acid. Okay. Oxaloacetate ialah keto acid. Kalau keto acid ni kita plus dengan N ataupun amino group, dia akan jadi amino acid. But if amino acid ni kita remove dia punya nitrogen ataupun amino group, dia akan jadi balik kepada alpha keto acid. So that's the concept or strategy that we're going to use, which is known as, I'm going to write right here, transamination. This one ialah proses dalam amino acid metabolism. Kita akan tengok dalam protein nanti. Konsep tu digunakan dalam shuttle ni. The same concept. So sekarang we need to convert or transform oxaloacetate into aspartate. Aspartate ni ialah amino acid. It's an amino acid. So alpha ketoglutarate itu ialah alpha keto acid. So apa kita nak buat? We need nitrogen. Kalau you look at the structure, sini, it becomes ada nitrogen pada aspartate. Bilangan carbon still the same. So mana nak dapat this nitrogen? So we have in the body, kita ada satu lagi amino acid. This amino acid, nama dia glutamate. Glutamate ni ialah nitrogen carrier. Dia boleh bawa nitrogen dan dia boleh donate. Dia boleh donate, sedekah lah. So dia akan beri dia punya nitrogen kepada oxaloacetate. It will give the nitrogen to oxaloacetate. This is known as transamination. The transfer amino group. So itu isi berapa? Tengok transfer isi berapa? Isi 2. So tengok enzim dia aspartate amino transferase. So what happened? Glutamate will pass the amino group kepada oxaloacetate. Now that oxaloacetate jadi apa? Oxaloacetate akan becomes aspartate. Alright. Okay. Oxaloacetate akan become aspartate. What happened to aspartate? Sekarang dia boleh boleh keluar ikut glutamate aspartate transporter tadi tu. So ni pun keluar dekat intermembrane space. Itu cerita dia. Sampai dia dah keluar, dia keluar dalam bentuk aspartate. However, kita aspartate ni of course lah, dia bukan oxaloacetate. So meaning that bila dia nak form balik into oxaloacetate ni, dia kena release balik lah. So use this formula. Formula yang sama. Sekejap, color lain pula. Biru lah, biru. Ni, formula ni. Guna formula yang sama. So sekarang because aspartate ni ialah amino acid, kita nak dia jadi balik alpha keto acid kan? So apa kita buat? Kita remove lah ni nitrogen ni. So dia boleh jadi balik oxaloacetate. So that it can form back into oxaloacetate. So in terms of removing, so amino acid remove nitrogen, dia akan jadi balik keto acid. Macam dia nak buat? Ada benda yang dia kena release, ada benda yang kena terima dia. In this case, the one that can accept dia ialah alpha ketoglutarate. This is also an alpha keto acid. Dia ni pun boleh terima juga. So aspartate akan pass on kepada dia punya alpha ketoglutarate. So aspartate ni akan jadi semula bentuk oxaloacetate. And then dia boleh pick up lagi. Itu cerita dia. But let's look at bila aspartate, itu satu Let's look at once aspartate donate ataupun remove dia punya nitrogen ataupun transfer the amino group kepada alpha ketoglutarate, apa akan berlaku? Alpha ketoglutarate ni akan membentuk menjadi semula kepada glutamate. Ni macam pindah-pindah lah, pindah-pindah rendah. Transferring. Ini semua topik amino acid metabolism. So dia akan jadi balik glutamate. Once jadi glutamate, so this glutamate can again function to supply the next oxaloacetate with dia punya nitrogen. So glutamate kena masuk balik. Kat mana dia nak masuk balik? Mari kita tengok beramai-ramai. Dia akan masuk balik ke dalam melalui this protein channel ataupun this protein carrier transporter tadi tu. Dia akan masuk balik ke dalam through this protein glutamate aspartate transporter through this channel. Dia akan masuk ikut channel ni. So dia masuk balik dan dia bersedialah untuk to donate dia punya nitrogen to oxaloacetate. Itu cerita dia. Okay. Kita pasal glutamate. But question, mana datang alpha ketoglutarate ni? Where does it come from? Oh, it actually comes from yang ni. Glutamate tadi, remember that dia donate ataupun dia transfer dia punya amino group kepada oxaloacetate. Once glutamate dah tak ada nitrogen, once glutamate lost dia punya amino group because of dia dah bagi dekat oxaloacetate, same formula. Glutamate will become alpha ketoglutarate. Dia kan jadi alpha ketoglutarate semula. So once dia jadi alpha ketoglutarate semula, this alpha ketoglutarate lah yang akan keluar. That alpha ketoglutarate will go out. Orange okay kah? This alpha glutarate akan keluar through this channel. Because inilah channel for malate and alpha glutarate transporter. Dan so ready to accept ammonium ion from aspartate. Itu je dia pusing-pusing. Itu cerita dia pusing-pusing. Lama juga ni dia pusing. Just for the sake of nak prepare oxaloacetate. Just the task of preparing oxaloacetate so that it can form into malate. Barulah malate ni boleh carry that hydrogen ion and also the electron into the matrix of mitochondria. And then form into NADH. So this hydrogen ni actually originate from yang ni. Bukan dia yang masuk. The hydrogen itself. Okay. Kat mana benda ni berlaku? It happens in the heart and liver. Heart and liver. Macam mana nak ingat? Heart and liver But in comparison, untuk that shuttles in the skeletal muscle and brain, ini ringkas, lebih ringkas lah. Bagi saya lebih ringkas. Dia hanya melibatkan a very simple step. That is known as G3P phosphate. Right? G3P shuttle Ini G3P shuttle. So apa berlaku? Sama juga. The same NADH that comes from the cytoplasmic. Dekat cytoplasm tadi tu. We're going to carry this hydrogen into the matrix of mitochondria. Tapi kali ni dia tak pass on pada malate ke apa? Pada oxaloacetate tak. Dia akan pass to DHAP. If you recall dalam glycolysis kan ada DHAP jadi D3P kan? The one that yang ada dua tu. Inilah bendanya. the same intermediate yang sama. So dia satu intermediate dia ada banyak function. So here DHAP once dia terima hydrogen ni, so it target on that functional group, dia akan membentuk this glycerol 3 phosphate. So they carry ni they carry hydrogen ni. So simple here because we have cytoplasmic G3P dehydrogenase. We also have mitochondrial, which is embedded near the membrane in mitochondria. Mitochondrial glycerol 3 phosphate dehydrogenase. Di situ ada enzyme link FAD. Ni. E itu enzyme link FAD, which will accept the hydrogen from G3P. Dia akan terima hydrogen tu from G3P. Now that G3P ni akan go back form into DHAP. So here when FAD terima hydrogen, of course dia akan jadi FADH2. Asal-asal NADH. But now because yang terima tu ialah enzyme link FAD, so dia akan jadilah FADH2. So sama, dia akan pass pada quinone and then yang masuk terus ke dalam ke ETC pada kompleks 2. Kompleks kedua FAD masuk dalam kompleks 2 and then the electron will be passed on dan seterusnya. That is what happened for NADH in the shuttle in skeletal muscle and also in brain. Ikut location. This one just to recall balik lah, kita pun dah tengok benda ni dulu kan. Kenapa ada 36, kenapa ada 38, it depend on the shuttle. Kalau Na malik aspartate tu of course equivalent dengan 3 ATP juga. Kalau dalam G3P dia akan equivalent to 2 ATP because of dia dah jadi FADH kan. FADH2 bukan NADH lagi. So that is how it calculated to that amount of ATP. Alright. Once dah semua dah masuk, kalau you tengok kat sini tadi, FAD2 tadi kan? FADH2 tadi. So this is where the enzyme succinate dehydrogenase. This enzyme, kalau you recall balik, is the same enzyme that involved in Krebs cycle. I'm going to click through real quick. The same enzyme. Here. Enzyme yang sama. FADH, FADH2, FAD. Benda yang sama berlaku. So it happens dekat-dekat. Benda tu berlaku yang sama. They share the same thing. Sama juga structure ni dipakai. Yang ni pun sama juga. The same thing. Benda yang sama. Benda sama pada ETC tu. Klik through balik. Benda yang sama. So for oxidative phosphorylation, those are the things. But for this one, nanti you can, my friend will teach you what's going on. Sama juga menceritakan tentang ATP synthase tu in part 3. You just can go and just pick the speed ataupun pick the point that you want to understand more on this electron transport chain. Alright. Quickly, kita ambil 3 minutes punya break, eye break, 3 minute eye break before we quickly go into the regulation. Okay, I'm gonna stop my video. You all ambil lah 3 minit sekejap bangun and I'm gonna mute myself. Okay, let's start. So it's already 10:11. So siapa yang rasa dia dah mata dah bateri dah habis, ya no problem. Boleh bersurai lah. Isi attendance tu. Isi attendance Siapa yang dah letih-letih tu kan. Tak payah nak berpaksa, tak tengok pun tak faham kan. Nanti tengok pun tak boleh nak fokus. So might as well you just go. Nanti you just catch up balik dekat video tu. Okay. So kita tengok kat regulation selalu kan kalau misalnya kata if you let's say that you want to venture into drug development, you nak buat satu antibiotic ke, you nak buat ubatan ke, you nak buat something that you nak venture from nature ke, nak tengok ke does that affect pada citric acid cycle ke, sebab one of the part that it will target ialah citric acid cycle. Like what I did, saya target electron transport system. So I target it as something that akan bagi, because I work with pesticides, saya akan make sure that it target pada certain protein complex, and then I do that biosensoring for it. So citric acid cycle regulation juga is part of like one of the most sought pathways kalau you nak go for drugs development. Sebab apa? Sekejap. Contoh macam ni lah Contoh macam ni, we know that naturally these are the three major enzymes that can become a control points. Dia ada control points dia. Control points mean apa? Itu yang dia target for inhibition. If you look back in our enzyme tadi kan, enzyme study dulu kan, we talk about inhibition kan. Dia pun samalah. Dia target enzyme tu. Macam ni, pyruvate dehydrogenase, dia target yang tu lah. Kalau misalnya kata in the body kita high of ADP and pyruvate, dia akan stimulate PDH tu. Dia akan stimulate PDH ni. PDH, it's an enzyme. Kalau misalnya kata we have high ATP, dia akan inhibit this enzyme. Dia akan inhibit this enzyme. This is an example that I showed masa kita buat discussion tu. I think ada juga benda ni dalam ni kan. But even though pyruvate cannot be transfer ataupun cannot be transformed to acidic CoA, kita kena ingat acidic CoA can come from other sources pun. It can comes from fatty acid. Tak semestinya it comes from glucose kan. Tak semestinya. So it can get the acidic CoA on pun boleh. Cuma I will not use that pyruvate from glucose to form into acetic CoA. So other than that, pada cycle tu sendiri, pada citric acid cycle tu, this enzyme, kalau kita tengok this enzyme ialah isocitrate dehydrogenase. So inilah bila when we have like production of ATP. Let's say dalam badan dah banyak ADP dah, we have abundance of ATP already. So we don't want to produce more NADH. Kita dah tak nak dah NADH. We want to slow it down ataupun to inhibit it. So apa kita buat? It will be inhibited. It will be inhibited. Isocitrate dehydrogenase will be inhibited by ATP and NADH. So this shows that it will stop the production of alpha-ketoglutarate and also NADH, and the other way around. ATP pun sama. Kalau ATP tinggi, dia akan allosterically stimulate this enzyme. Untuk alpha-ketoglutarate macam mana? Dia ada enzim juga, alpha-ketoglutarate dehydrogenase tu. Ini sama ni pun sama. It will be inhibited by succinyl CoA itself. Substrate, ni substrate dia, ni produk dia. Produk dia kalau tinggi, dia akan inhibit balik enzim tu ni. Produk dia tinggi, it will inhibit this enzyme so that don't produce more of me. Stop producing me. Gitu. Apa lagi? NADH and also high ATP. We don't want to produce more NADH. That's the bottom line. So that is how they target, the body target kat mana dia nak regulate kita punya pathway. So more or less like looking at energy lah. Macam tadi kita tengok glycolysis kan. You look at the energy. If you have like high energy, we're not going to produce more of NADH. FADH tu dia tak produce lah. So kita control kat situ that is how the body regulate. Ni sama yang dia tunjuk hari tu. Okay. That is one part of it. Another part of it ialah oxidative phosphorylation itself. these are 4 major enzymes that regulate oxidative phosphorylation. Dulu saya target yang ni. Coenzyme Q. Dulu saya target yang ni. NADH, coenzyme Q reductase, cytochrome C, and glutathione reductase. These are enzymes that control the process of oxidative phosphorylation. Okay, dia pun sama. It's regulated for the need of ATP. If we need it, kita akan stimulate. If we don't need it, kita akan stop it. Other than that, to regulate ETC, kita boleh guna drugs. Bukan dadah berbahaya kan nyawa tu. Ni drugs is ubatan. You can use poison. I use poison. Last time I used poison. And also you can use chemicals. You can use chemicals to inhibit or to accelerate the activity of ETC itself. Contoh macam ni. Yang ni ialah kita tengok ada kompleks 1, kompleks 2, kompleks 3, kompleks 4. So contoh bukanlah ini this for nice to know. Contoh kita ambil carbon monoxide, cyanide. So these are all serious poison where dia boleh inhibit kompleks 4 kat sini. So is the complex 4 is inhibited, so be no production of ATP lah. ATP takkan terhasil lah macam tu. There should be no ATP. Kita block dia kat situ. Lagi apa? Malonate ni. Dia inhibit complex 2. Mycin ni semua antibiotik. Dia boleh inhibit kompleks 1. Macam tu lah. Ni ada lagi anti-mycin A, dia inhibit kompleks 3. Inilah when we identify what are the enzymes, kita boleh create drugs atau a poison that can stop that enzyme from functioning. Either dalam bentuk kompetitif, it can be bentuk uncompetitive, it can be bentuk non-competitive. We have to study apa behavior kita punya inhibitor tu. If you want to go for drug development, yang kita nak tengok. Or you can get it from a plant. Contoh apa nama pokok yang kat Ipoh yang tumbang tu? Lupalah pula nama dia. Yang kat Taman DR tu, yang pokok besar tu. Yang selalu kalau misalnya kata orang asal, dia suka guna dia punya getah tu untuk memanah, untuk menyumpit haiwan. Aduh, lupalah pula nama dia. Tak apalah, sekarang ingatlah. So that poison tu dia boleh inhibit, dia ada toksin tau, dia boleh inhibit the body function. Sama ada, saya tak ingat pula ada inhibit apa. Tapi the fact that if you want to study natural product, kita tengok dia punya behavior, identify the structure, identify protein, ikut protein data bank yang tadi saya tunjuk tu kan. So tengok sama ada dia boleh bind ke tak boleh bind. Kalau ada structure yang kita ada tu boleh bind pada enzim tu, berarti it's a good potential. Dia potential drugs. Dia akan jadi potential drugs. Sama juga macam the one that we did for marijuana kan. Kita test untuk neuron ataupun untuk receptor for epilepsy tu. So dia boleh bind. Sebab tu dia boleh reduce epilepsy. But bukan yang THC yang high tu bukan. Sebab orang banyak salah sangka, oh ambil ganja, ni boleh ubat. Tak. Sebab dia bukan semua yang tu yang perubatan. We are talking about that CBD, CBG, those are molecules yang boleh, yang ada drug potential. Bukan drug dadah tu. I mean like for medicine lah. So if you understand pathway ni, you can understand macam mana kita boleh target dia punya ubatan lah gitu. Okay, ini pun sama juga yang tadi just a different way of looking at. For plant and animal, more or less the same saja except for the location. Animals happens in intermembrane space. For chloroplast, the thylakoid. And then for all those production of ATP happens in matrix. Untuk plant dia berlaku pada stroma. So itulah perbezaan. Dia cuma dia terbalik sikit lah nanti. Nak faham dia. Okay. Regulation dia macam mana? Regulation, apart from having enzymes to regulate, we can also regulate kita punya activity by the hormone. Ini contoh hormon yang terpenting. Insulin and also glucagon. If you look at the red arrows, itu semua maksudnya dia inhibit ataupun dia slow it down. Yang the green arrows, ni semua ialah dia stimulate. It stimulates all the processes. If we pick one, if we have glucagon high in our blood system, it will start stimulate protein catabolism, breakdown of protein, ataupun ureagenesis, forming urea, and also ketogenesis. Ni kalau misal, ni kalau misal kata kita ada search of glucagon in the body. Apa lagi, glucagon juga boleh help in promoting glycogenolysis, break down glycogen. Bila glucagon tinggi, berarti dalam badan kita dah low of sugar kan. So kita nak gunalah sumber kita punya simpanan kita tu. So that is, dia akan go for glycogenolysis as well as gembira-gembira di sini, lipolysis, break down the fat. Insulin on the other hand, kalau tengok, kalau misalnya dia tinggi, dia akan promote glycogen synthesis, lipid synthesis, as well as protein synthesis. You can go go around and look at how it connected to one pathway to another pathway by looking at the arrows. Okay, I'm going to leave that one to you, and you can look at it after this.